So yes, I'm going to tell you just what we've been doing, trying to look exactly using patch clamp to understand circuits. So it's certainly electrophysiology, and of course everything's ion channels, but more on the electrophysiology than ion channels today. So what pretty much I just introduced because I want to tell you why we're doing these things and why I think the questions that we're asking are interesting for me. And that is because the way I think about the brain as a very simple man is that everything that we know or have experienced or want is very simply just activity passing from some set of neurons to other neurons around inside your head. That's how I see the world. And therefore, everything that you've ever wanted and everything you've ever hated is just simply activity passing from different subsets of neurons to different subsets of neurons. And of course, it's an oversimplification. But when you see it like this, the question for me is simply, how does activity pass from specific subsets of neurons to other? And what is the properties of transmission, and how does this give rise to computations? And so where we're looking is the hippocampus, which is one of the, must be one of the least studied brain areas in ever, of course. And so everyone will know about the hippocampus. It's a sausage. It's a sausage in the middle of your brain. Oh, sorry. A sausage in, can you see my pointer, right? Oh, yeah. So the sausage in the middle of your brain here. And if we cut a slice through, it's this classical circuit, which if we're to reduce it to the simplicity, it takes information from the entorhinal cortex and gives information to the entorhinal cortex. And in some way, this circuit loop can store information. How? That's sort of the question. But in some way, activity passing around this loop stores information. And we know this because if I was to take out both of your hippocampi right now, you'd probably not remember very much forever. And so the question for us is, how can this circuit, using cells and synapses, store stuff? And we're particularly interested in this area here, which is CA3 and the dentate gyrus. And the reason for that is if you just break it down to the simplest theory, you've got a really big bank of dentate gyrus granule cells, and they wire into CA3. And CA3 is not like CA1. I mean, it's a little bit more complicated and interesting because it has recurrent connections. So you have synapses between the pyramidal neurons. And what this means is that information that flows into the system, activating granule cells, might represent perhaps different parts of an experience. But because all this is going to get put together by recurrent network, this CA3 network is able to store and associate different parts of your experiences as memories. That's the sort of idea. And so for us, the question really is, what is the arrangement of this network? How does this recurrent network, what's the properties of the synapses, of the cells, and how does this manage to do what it's doing? So that's where we sort of begin. And as I've already alluded to, what I find really important is simply understanding how activity passes from individual neurons to individual other neurons. And hopefully in the next few slides, I can tell you why, give you an idea of why that's really important. And that's because even if we take this simple here, you have granule cells wired into CA3, a recurrent network. If you were to arrange the synapses completely randomly, if you were to arrange them in clusters, if you were to wire inhibition into some cells or other cells, or for example, if each neuron had completely different properties, every single one of these different changes would change how activity passes through this network. On a simple logical step, activity will pass in different routes, in different ways, in different patterns. And therefore, what we want to do is understand the shape of the circuit from the level of individual neurons. And I think that's very important. And so how do we do this? We do this using multicellular patch clamp. So of course, we take acute hippocampal slices, and we have this setup here where we can record from 8 neurons at the same time with patch clamp. And so you can see here, what we do is we're going into the acute slices and trying to record as many cells as possible. And sometimes it's 8, sometimes it's slightly less, because of course, every one of you who's done patch clamp will know that not everything in life is perfect. But that's the principle. And I think it's a really powerful technique because it gets you really detailed information about cells and all of the details of the cells, every cell you've recorded. it gives you the properties of synapses between the neurons recorded, and you have the perfect control because you can stimulate presynaptic cells and generate action potentials and record postsynaptic currents. And therefore, for synaptic transmission, it is unitary in its nature. You're studying the properties of one synapse. And of course, because you've got 8 cells at the same time, you can study circuits, local circuits to some extent. So you can look for which patterns of connectivity are happening more than others. And of course, you might ask, why do we do 8 at the same time? And of course, it makes life very painful. But the point is that if you do pairs, you are testing 2 possible synaptic connections at once, whereas every single pipette you add is massively increasing the number of connections you're testing. So when you're testing with 8 cells, you're testing 56 possible connections between neurons. So every pipette really increases the amount of throughput for getting data and getting study of the local circuitry. So that's our technique. This is my form of electrophysiological torture, and I think is quite interesting. And so I'm going to give you just a sort of a look at 2 projects we've done. The first one is looking at the heterogeneity of cells and discussing how different properties of different cells and taking into account heterogeneity can give you interesting features of the local microcircuit. And the second thing I want to tell you is about how we can try and relate this to the human brain, which is, as you can imagine, a little bit more difficult. So to begin with, mice. So this is now mouse hippocampal slices. And as I told you, we're trying to understand CA3. This is a recurrent network. And here you can see one recording I've done. Here's a handful of cells that we've imaged after the recording. And they're all CA3 pyramidal neurons. But from the literature, the historic literature, there are a lot of different properties of pyramidal neurons that show differences. Some cells have different, firing properties, morphological properties, and we can see these differences. And therefore, because we're doing our recordings, we have to take this heterogeneity into account. So here, for example, we can classify our neurons into 2 types of neurons. One, which are deep, and those that are superficial in the layer, as has been seen in CA1, and follows what other people have seen, that we can see differences in the morphology and the somatic location and things. And so the question for me with heterogeneity is, I think it's very interesting because there's obviously secrets in this heterogeneity, but it's obviously very difficult to understand, because it's never clear what is ends of a gradient, what is meaningful, because of course every single individual neuron has a different shape to every other neuron, and going to that level, you don't know what is actually relevant and important. And so what we try to do is simply look at the properties of the neurons and ask whether or not we've got different classes of cells within this heterogeneity or one population. And so the first thing that we do when we're recording our neurons here is just inject current steps, as everyone do when you're patching neurons in slices. And here you can see a perfect example of a deep CA3 neuron, which gives you a burst activity upon current injection, and a superficial neuron which doesn't. And so plotting this is simply just injecting more current, and you can see the frequency of this initial burst here on the y-axis. And what you can clearly see is that the 2 types of neurons classified by morphology fall clearly into 2 populations. And so for me, this example showed me that what we're dealing with in CA3 is not one big pile of CA3 pyramidal neurons. There's at least 2 subpopulations with differences based purely on the burst frequency. And then obviously we can also plot this against somatic depth, because I showed you already they have different somatic depths. And what you can see is burst frequency against somatic depth gives you complete segregation, well, almost complete segregation of 2 clusters. So, I mean, in general, in the brain, interneurons are the stars of heterogeneity with lots of different cell types. But more and more these days, especially with genetic techniques, there is the idea that there is populations and diversity within excitatory neurons. And I think it's a really interesting thing to explore, and it's really emerging now, is looking at these properties of these different cells. And here, what is important is we can separate them morphoelectrically into different subpopulations. And the most important thing about being able to classify is that you can then look at distinct properties of the 2 different cell types that you couldn't see if treating it as one population. And so with this information, we can see different types of pyramidal neurons based on their firing. And a long story short, I'm not going to go into, there is differences in the amount of input the different cells receive. There's differences in the intrinsic properties of the cells, and loosely, without too much more details, superficial cells receive more input, but deep cells are wired or built to give more output when they do receive input. But the main thing that we can do here is, of course, look at the connectivity between the neurons. So we record a big pile of octuple recordings from across CA3 and look at exactly how often we found synapses between the cells. And of course, CA3 is a recurrent network. So between these superficial cells, we see around 4% connectivity. We can see connectivity from these superficial cells into these peculiar deep cells. We can also see connections between deep cells, so they're synapsing onto one another. But what we saw like very little of was connections from deep cells back to superficial cells. And so what this means for, like, I guess memory storage and things, let's not even worry about. But the first thing to say is that if you can break heterogeneity down and understand the differences in the data you're looking at, there is secrets and there's interesting features of circuits that you can see within that. And so this is sort of my evangelical preach to explore any heterogeneity you have, and this counts for any neuroscience data set, because there's interesting information there. And so the next thing we did actually is to look at these different types of cells, is look just at the recordings, and here what we can see is 5 cells that we've recorded, 2 superficial and 2 deep. And this is my second evangelical preach, which is to advocate for staring into the abyss of your raw data, because analyzed data tells you very little, but raw data is where you can see insights yourself with your own eyes. So if we look at things here, what you can see is spontaneous currents. These are just synapses. It's a mixture of excitation and inhibition because of our internal solution. And you can see synaptic input onto our recorded cells, and we can detect these events. But what you can also see is that in certain places, for example, down here, you can see 2 cells that are receiving input at exactly the same time. And this is quite interesting. And I'm going to go on a 1-minute tangent just to warn you of demons. And this is just to say that, so we are looking here at spontaneous activity, and I will tell you what it means in a minute. But when we've done some work looking at detection of events, and this is actually a little bit of a simulation study, just to look at how detected events in minis and in spontaneous events, spontaneous current recordings, represent real changes. So what we did is just look at events that we're simulating here, and we could increase the amplitude of events. And you can see here that we're adding 2 picoamps onto each event. And what we find is that if you increase the size of these events here, we can see a shift in the distribution because we're increasing the amplitude. But when you look through the detection, through event detection, we see a very different distribution. And the reason for this is because small events, as is pure logic, small events are lost in noise and you cannot see them. And everything I'm telling you here and everything we did in this study is actually just pure logic, just to say, of course, small events, when you're looking at synapses, when you're looking at any data that has a threshold and a detection threshold, will be obscured. And, I guess what's the word? You'll look through the prism of detection and you'll see things in a different way. But I just want to tell you that here, what we've done is a simulation study where we're increasing purely the size of synaptic currents. And what we do is, by detecting, we actually detect no change in event amplitude and pure changes in mini frequency. And so all this is to say that I hope everyone can be extremely careful of looking at the quality of data when you're analyzing spontaneous currents and mini data, because the interpretation of your changes are probably going to be not what you first think of. And you can see this data is on bioRxiv. I'm happy to discuss it another time, but it's just a warning that anything that's close to the detection limit, and that means synapses, is difficult to interpret. So back to our events. So what we've done is we've checked, because of this study, we've checked that, of course, our noise levels are the same between all our conditions, so we can see our events in a reliable way. And when we see these correlated events, what they seem to tell me is that probably these two cells have a presynaptic cell that's providing input to two of our recorded cells. And so, of course, it looks like it's occurring here. And so we analyze this, and you have got to analyze it to get the numbers on it. And so what we do is we plot all of the time difference between one event in one cell and the events in every other cell. So you get sort of these histograms of time difference, delta t. And this is a histogram of all the events in cell 1 and 2, as I've shown you, with a bin width of 1 millisecond. And what you see is a beautiful peak purely at time equals 0. And therefore, what we have is a population of synaptic input that happens at exactly the same time on cells 1 and cell 2, which seems to be probably the same presynaptic cell population. And we can do the same for other cell pairs. And we can look here at cells 4 and cell 5, which are both deep cells, and we can see this beautiful peak again. But if we compare cells 1 and cells 5, we don't see anything. And so putting this data together, we can actually look for cells that seem to be embedded in the same local circuit. So here, what you see is pooling many recordings, and you were looking at the correlation between events of the same type, both superficial cells or both deep cells. And it's clear here that what we see is that this shared input, the shared synaptic input you get just by looking at the event correlation and this correlation of spontaneous activity, you can see is shared within cell class, but not really at all between cell class. And we can actually put on gabazine, and we can block the correlation. So we still see events, we see a lower event rate, of course, but all of these correlated events are lost. And therefore, what we believe this tells us is there are specific interneurons that are wiring either into one cell population or the other cell population. And this is quite interesting because for the circuit level, it means that, as I've just told you, there's these two different sort of recurrent sublayers within CA3 that you see from the heterogeneity, and that interneurons are able to potentially gate which of these two layers are active. And so this is sort of a little breakdown of what we've done in mice. And the main point of this is not to say, look, these are the features we've seen. The main point I'd like to raise is that by looking at neuroscience at the level of single cells, you can see things that you wouldn't otherwise have seen when you look at populations and all sorts of stimulating large populations of cells. And so looking at single cells is important. So that's it for mice. And so this is how I see neuroscience in the mouse, and you can call it a little controversial, but all we need to do to solve the brain is to determine all the different types of cells we have in the brain and their activity, who's connected to who, and what they're firing like in vivo. And we can do all these things with current neuroscience techniques, right? And if we put all these pieces of information together, you're going to be pretty well on the way to solving the brain. However, the question that we still have is what about humans? Because none of our neuroscience techniques we can apply very well to human tissue. We cannot make transgenic humans. We cannot do viral injection to humans. We cannot do many of the sort of neuroscience manipulations on human brain. And so what we've been asking ourselves here is, how do we even understand human tissue? How do we understand human circuits? And so that's what I'm going to tell you about for the rest of this talk. So this work is my second bullet point. And so this is a collaboration that has started with the Medical University here in Vienna with Professor Karl Rösler. He is the head of neurosurgery there, and his is one of the most fantastic collaborations I've ever been involved in. And so he's performing surgery on human patients for epilepsy. So in epilepsy with temporal lobe epilepsy, you take a tissue block of hippocampus from where it seems that there is a seizure focus. And so what this means is that we have access to pieces of human hippocampus that will be removed from humans and is available for research. And therefore, we can use this to study human tissue and human circuits with the same resolution, with the same techniques as I've just showed you in mouse brain. And so just as a little bit of background, what we're doing is we're driving to Vienna, and this is where the hospital is here in Vienna. And we're out here in the forest, which takes about 35 minutes to drive. And so what we've designed is this little transport box with a carbogen container in, and this has been supported by many people here at the Institute. which allows us to take these pieces of tissue immediately as soon as they are taken out of a patient brain and take them out to the forest where we can make slices. And this is a project that I did in collaboration with Victor and Rebecca. So we've been recording together, through the night, as you need to do when you've got these very valuable samples. And so here's a few pictures of what the tissue samples look like. This is a block of human hippocampus on the slicer, and here's a few slices. So you can see that the size of the human hippocampus is larger than even a whole brain slice from a mouse. And the first thing to say is that, of course, every single thing that we're studying comes from epileptic patients. And of course, everything could be an artifact of epilepsy. However, at the end of the day, this is the first view that we can have on human circuits and human tissue. And therefore, it's even with that caveat, it's very interesting to explore the properties of these cells because, the epileptic human is probably a better model of a human than a mouse is. So we get two types of samples. We get those that look like this, and you can see the hippocampus in its beautiful interlocking C shapes. But you also get samples that look like this. And this is hippocampal sclerosis. And what you have is granule cells start to move around because of the disease. And there is a lot of loss of neurons in different subfields like CA3 and CA1. And so these two different types of samples, if we put it bluntly, give you a view on physiology in one hand and pathology in the other. And you can look at both of these two different worlds with these different samples. Everything I tell you today is going to come from these non-sclerotic samples. So they're still epileptic, but there's no loss of neurons. The structure of the hippocampus looks intact. And therefore, it's the closest possible view that we can have on human cells, human circuits. And so that's what we talk about today. So the human brain, as I've told you, is quite large. Here's a human hippocampal slice. Here's a mouse hippocampal slice for reference. So you can see we're talking about very different scales. And just to orientate you, this is the dentate gyrus here, this beautiful ribbon of neurons. In green, we're staining all the neurons in the slice with new N. And here you can see a small cluster of neurons that are my biosite and labeled cells in CA3. So we put together many recordings from many patients and try and characterize exactly the same again, the properties of CA3, neurons, and circuits. And the question is, how can we store information in this recurrent network, this time in human brain? So the questions here are, how do human hippocampal circuits support memory? And are there specific properties that we can see in human tissue that we don't see in the mouse brain? And so the first thing, I'm going to answer the second question first, simply by showing you a few more of these current injections. So what you've got here, we're just injecting currents into a mouse neuron and a human neuron. It's not the practical to have a question and answer here, but the question would be, what is the difference you can see here? The difference is very obvious. You can see that the precision, the interspike interval is very, very, very thin in human brain, but not in mouse brain. And therefore, interestingly, why? We're not sure, but there clearly seems to be precision to spiking activity in human cells that we do not see in mouse cells. The second thing is what we can look at the properties of synapses. So we're looking now at the properties of synapses by stimulating one cell. Everything is pairs, one cell, and recording from another cell. And we search for these synapses while in the tissue. And you can see in CA3 versus in the cortex, in layer 2-3 of the cortex, these are different synapses that are doing different computations. And we can clearly see that the CA3 synapses are slower. So the rise time and the gate time of the EPSPs here are much slower in CA3. And therefore, hippocampal synapses in the human brain do have the same slow integrating properties that you would expect from what we see in mouse, which is interesting. And of course, this is probably related to their function in association and memory storage. But if we now compare our synapses to mouse synapses, this is an example of pairs in CA3, where we're stimulating one cell and looking at the response from another cell. In mouse, you can see, as many of you who have looked at synaptic transmission will know, that you see difference in the size of responses. You see failures, often in failures of transmission. But what we saw in the human brain was very different. We saw very reliable and very precise synapses. So we saw very rare failures, which you can see in the success probability here. And you also saw that the variability in the size of these responses was much lower. And so in short, we see precise spiking and precise synapses in human tissue. And therefore, it seems to be that human circuits will be working in some different way to what we would expect from all the physiology that's been done in mice. And now to move on, I'm going to spend the remainder of the talk talking about synaptic connectivity and what we can learn from our experiments in slices about the circuit architecture and what I think that means for memory storage. And so we've done our experiments taking pieces of hippocampus here. And here's an example. We do our octuple recordings. And here you can see what we're doing is simulating each cell in turn and looking at responses from other cells. And what you see here is actually no synaptic connections between the cells. And that's more often than not exactly what we saw in the human tissue in hippocampal CA3. And so the first thing I always would ask myself is, am I doing my experiments wrong? Am I doing my experiments badly, or is the tissue quality not very good? And so what we can also take is samples from the cortex, because the surgeon has to remove pieces of cortex to get to the hippocampus. So they will take out a piece of cortex to get to here in the hippocampus. And so we can do our same experiments in the cortex. And the cortex has been much more characterized, much better characterized. So you can see here a cluster of neurons that are recorded in layer 2-3 of the neocortex. And here what we can see is many, many, many synaptic connections. So this cell is connected to this cell. Here you've got a synapse between these two cells. And so this gave us the assurance that what we're doing is actually nice experiments with high quality tissue, but there's actually just low connectivity in CA3. And so if we quantify what we see, we actually see that the connectivity in CA3 here is 10 times lower than what you see in the neocortex. And if we put this together with what we see across species, we actually see a decrease in the connectivity between CA3 neurons from mouse to rat to the human brain. And so this is actually quite interesting that we see this sparsification of connectivity, because in the neocortex, you see totally the opposite. In the human brain, people report much higher connectivity between neurons, lots of dense synapses between the cells, and they say, oh, look, this is why the humans are smart. You know, they've got loads more synapses, lots more connectivity. But here in CA3, we see the opposite. Why is that? And hopefully in the experiments that follow, I can convince you that one, it's interesting, and two, it's meaningful for circuit computation. So can we make sense of this? The first thing to do is look at our cells. This is an array of mouse neurons, mouse CA3 neurons, and here's an array of human CA3 neurons. And the first thing to say is human neurons are larger. We also wanted to look at the synapses, to look at the input and the arrangement of synapses on these cells. And so, sorry, we're going to turn away from the patch-pipette for a moment, and we're going to do some imaging. And so this is a collaboration we did with Hans Danzel and Mo Tavakoli, who were downstairs in this building. And it's really lucky that they were inventing techniques, and they're using techniques such as expansion microscopy, because it means that we can take pieces of human tissue and we can expand them and look at synapses. And so this here, what you see here is a piece of jelly that used to be a piece of human brain. And it just looks like jelly. But if we look inside this jelly, you can image the structure of dendrites, of synapses, of axons. And so the technique that they've invented, I put the QR for the paper here because you really should read it, is effectively using confocal microscopy and expanded tissue is connectomics that is moving towards the level of what you can do with EM, but just sitting on a confocal microscope. And this is really quite incredible. And because they're here, we could use this and apply this to our tissue to understand how the circuit is arranged in our human and our mouse brain. And so this is actually work that was done. I had Rebecca's face here. She was a rotation student, now a PhD student here, who was looking at helping to reconstruct inputs. And so here you see two dendrites, a mouse dendrite where we're reconstructing inputs. This is multi-fiber input or a human neuron. And so you can effectively do mini connectomes using this technique. And so I've already told you that mouse neurons are smaller than human neurons, but because of our super resolution microscopy, we could measure the input, the spine density on these two different types of cells. And we see that mouse neurons, surprisingly, actually have a spine density that is twice as high as human neurons. So this is mouse neurons here, you can see human neurons here. So the spines are just further apart. And this actually has been seen in EM connectomics in the neocortex as well. So probably it's a feature of the human brain that synapses, excitatory synapses on spines are more separated in human tissue. Why? Again, it's an interesting question. We're not sure, but it seems to be a feature. And so what we can do is to start understanding the circuit, we can do some very complicated modeling, which is called mathematics and multiplication. And so we multiply these two numbers together, how much cable a cell has and the input density, to get estimates of how many inputs we have per cell. And so what you see is actually, surprisingly, despite human neurons being twice as big or even 3 times as big as a mouse neuron, the number of excitatory inputs each one has doesn't seem to be that changed. And this is quite interesting because one question that comes to me at conferences even last week was, look, we look at a mouse brain, we study a mouse brain, and it's going to be exactly the same. It's just a human's big mouse, no problem. But what we see here is that different features of the circuit are scaled in completely different ways. So CA3 neurons, actually, this is not what we've done, this is from the historic literature, have gone up by about 30-fold the number of neurons. The inputs per cell is decreased, as you saw, the spine density is decreased, the number of inputs on the total cell is similar, connectivity is decreasing in CA3, increasing in cortex. And so the point is, the takeaway here is that the human brain is not just a big mouse brain. There's lots of different scaling factors that you have to take into account. And that's important if you want to understand really human circuits. So what was quite interesting from this is looking at human tissue, we see that the human brain has expanded in size, of course, but mainly by expanding the number of cells in CA3, this is, rather than increasing the number of inputs per cell. And this is quite an interesting question as to why a brain would say it wants more cells rather than more complicated neurons. Because in theory, if we were to take a small recurrent network, let's call this a mouse network, and make it bigger and more complicated, there's two ways you could do this. And we could take two theoretical humans. One could have really dense connectivity. So you could make neurons super complex with millions of inputs, millions of synapses, or you could have the same number of synapses as this dense network, but distributed across many cells. And so what we've done is to do a simple theoretical modeling experiment to say why, when expanding the number of synapses in the brain, from mouse to human, has the human brain chosen this route of more neurons? And so this was work done by Andrea in the lab here, so simply using a simple theoretical model of networks to store and remember patterns in this network. And what we saw with this is that it doesn't matter how big your theoretical recurrent network is, it's always going to have a higher memory capacity when you arrange it into more neurons and sparser connectivity rather than less neurons with many synapses and dense connectivity. And therefore, I'm not saying that this is exactly why, but it's interesting to speculate that when you want to expand the size of a brain, or where you allow size expansion in brain, storage capacity increases with more neurons, and that's better to prioritize than more complex neurons. So now, because we've done all this anatomy, you're probably wondering why I'm doing all this boring anatomy, we can try and understand the circuit. So by taking the number of inputs per cell and dividing it by the number of cells in the network, you get a measure of connectivity. It's simply saying, if there's 100,000 cells in a network and I can have inputs from 10 of them, that tells you the percentage connectivity in the network. And so when we do this, we can purely use anatomy, and we know that the number of cells in each of these networks, and we know the number of inputs per cell, and we could predict that for a pure randomly connected CA3 recurrent network, the connectivity would be around 6% in mouse and decrease massively in the human brain, because the human brain has expanded number of cells and not the number of inputs. And this actually is very interestingly super paralleled to what we see biologically measured using multicellular patch clamp. And what does this tell us? I would propose that what this tells us is that actually CA3, when we patch it and when we look at it locally, actually reflects the fact that CA3 is behaving as one big network, just like our theoretical anatomically calculated network. So CA3 is behaving as one big network. And therefore, the sparsifying connectivity we see across species, different to the neocortex, reflects how the circuit is shaped. And so the hippocampus is shaped like a large recurrent network, and the neocortex is more clustered into dense local columns or computational units. And therefore, when we do our recordings, we are recording from small clusters of local neurons, and we're likely to see no or less synapses versus these dense units. And therefore, so by recording with our multicellular patch clamp, even in slices, we can see this structure of the circuit for the hippocampal CA3. And so here what you see, this is going back to our schematic, what we will propose is this sparse connectivity in the hippocampus in CA3 gives you a network that is able to associate stuff. And so it's able to associate all sorts of incoming input, which is exactly what you want from an associative memory storage network. And so that's the sort of conclusions that I take away from this work is that brain scaling is not simple, that maybe we should think a little bit more about what the properties of human synapses and circuits are, because I think most people, me included, I'm classically a mouse physiologist, would walk around saying, yes, we're trying to understand the human brain. But to understand the human brain, we have to at least take a little look at human brain tissue. And so I just want to leave you with one last provocative slide, and that is to say, we return here to our array of recorded neurons. These are all my biocytin-filled recorded neurons. And you can see that the human brain has this incredible complexity that you don't see in the mouse brain. And so we can see cells that have one apical dendrite. We can see cells that have two apical dendrite in both species. But in the human brain, we can also see massive extension of basal dendrites, these so-called taproot dendrites that have also been seen in human cortex that we never see in the mouse brain. And we can also see cells like this that are inverted, flipped with respect to how you'd expect the apical and basal dendrites to be. And not to go into this any further, just to say that the complexity of neurons in the human brain is going to be even more than the mouse brain. And I've already given you the sort of introduction at the beginning that the cell types in the mouse brain we're still only just exploring. And so the way I sort of see neuroscience is a little bit like this, whereby we are already dealing with the complexity of the mouse brain, and we're trying to deal with understanding the complexity of the mouse brain. So what are we going to do to understand the complexity of the human brain, which is a much greater challenge? And that's a question I'm happy to discuss with any of you. And so this work has been done as a really nice collaboration with the lab of Peter Jonas here and the lab of, the neurosurgery, Carl Rössler's neurosurgery in the city center. And there's a lot of other people who've been involved for recording and patching, and it's been a super collaborative project. And so I'm very grateful for working as part of this very nice team. All of the papers can be seen as papers or preprints here along the bottom, so you can have a look. And if you want to discuss, please ask me now or send me an email if you fancy it anytime. And so hopefully there was something interesting for you there, and I'm happy to discuss.
Fantastic, Jay. That was a wonderful talk. Thank you very much. So many cool ideas there. I'd encourage anyone, if they have questions, to add them to the Q&A box now, or if you want to raise your hand, we can unmute you. We've got one question in from Damien Bell, who says, octuplet recordings. Wow, that sounds tortuous. Congratulations and great work. Taking heed of your demons in every detection and correlation. Just lost it. Coming back. Taking heed of your demons in event detection and correlation. Your 1 millisecond bins of event correlations would likely be affected by axonal distances between putative connected neurons. Do your event correlations allow for distances that may cause correlated events that lie outside of the 1 millisecond bins?
I mean, we look at the histograms and what we see is as you can see, the bin at 0 milliseconds, this is plus or minus 0.5 milliseconds, is way above the noise level. And we see some increase in the second bin, but it was not very large. So, I mean, practically, because of the structure of the data, we just consider a correlated event with plus or minus 0.5 milliseconds, because that's where we see things coming above noise. I mean, it's actually an interesting point, and it would actually be interesting to look at the shape of these peaks above noise and whether or not different synapses or different inputs show more jitter, I guess. So, I mean, I guess it's another way I can analyze the data. So maybe I can take a look at this. So thanks.
Rian says in answer, or to build on that question, actually, could they apply distance adjusted latency corrections? Maybe you covered that in your answer.
Well, I mean, the thing is, so when we're looking at this, there's two things that we don't know many things. What we're looking at is simply, this is just one neuron. We're looking at all the spontaneous input to this cell. So they could be happening at any point on the dendritic tree. And also the distance from the other cells, we also don't know. So we don't know how far the input has come. Actually, what I think is that from everything else we've done, I think this is probably most likely fast spiking local interneurons, probably PV interneurons, which are have very tight timing of their synaptic transmission, as you will know, and also close somatic location. For longer range connections, things would be more complex, yes. But for this exact effect that we see, I think that the fast timing of interneurons and these fast spiking interneurons mean that it's not going to be so much of an effect on these cells, especially as the input comes perisomatically as well. But yeah, we have to think.
Another question here. Can you explain the effects of gabazine on the detection of correlated inputs to pairs of cells in mouse CA3?
So pretty much with the gabazine, what we're doing is, so it sort of looks slightly confusing because all of our spontaneous inputs go in the same direction, and that's just the balance of our ions means that we record inhibitory and excitatory currents going in the same way, purely due to the technical setup. And what it means is when we do the gabazine, we can block the inhibitory component, and when we apply gabazine, we block the inhibitory component, we see complete loss of this correlated events. And therefore, what I would explain from this is that it tells me that the correlated input, these shared input into two cells, is coming from an inhibitory neuron population, a GABAergic population. Yeah.
I had a question on the differences in the human and mouse success of the excitatory synaptic inputs. Sorry, I'm not a neuroscience, you might be able to tell. I wondered whether you would see the same thing for inhibitory responses, and maybe given, as you say, these are epileptic brains, does it reflect the imbalance of EI imbalance in human samples that isn't present in the mice?
Yeah, well, we haven't looked at inhibitory synapses. I would love to look at inhibitory synapses. The problem is that there's obviously we have limited samples, and also there's only 24 hours in the day, sadly. But there's many of these things that would be really nice to look at. In terms of the reliability, this has also been seen in the neocortex as well for synapses by the team in Amsterdam. So it perhaps is a feature of synapses beyond excitatory synapses. And I can't remember what the second part to your question was.
No, I think that's right. My question was getting at whether the increased success rate in the human samples might reflect an EI imbalance because of epilepsy, right?
I mean, whether it's an effect of the epilepsy or the treatment is totally possible, of course. And the point would be, we, and unfortunately, cannot at the moment have wild-type human brain. No one is giving out wild-type human brain, and this is the best possible we can study. We would love to also have samples from patients where they have tumors near to the hippocampus. These are available rarely, and that would allow you to look at non-epileptic brain tissue, and that's where you're going to get the answer. And so we will do this if possible, but it totally depends on which patients and which samples are available to us. Yeah, but of course we have to think about epilepsy.
Following on from that, John Gargos in the chat asks, were any samples from monogenic, I think that's schizophrenia disorders or SZ disorders? Would that be schizophrenia? Or maybe it's... could you speak more broadly about the genetic? Oh, sorry, seizure. Sorry, of course. Yeah.
Yeah. No.
Could you speak more about the genetics or the age of the samples that you received?
Yeah, the age of the samples is from 15 to I think 59. So it's a very broad age range, and we've not seen any difference in properties across age. In terms of the genetic background, I I don't have any information on the genetic background. It would be very nice to map this to see what is, genetically caused. I think, as far as I understand, we don't have that information here. But of course, more information on the patients and the details of the epilepsy would be super nice. And to break down any of our information and measurements into patient individuality or human individuality would be an interesting angle, but we don't have that information. We have quite limited data here.
I have a quick question. So because these are epileptic samples from people with epilepsy, I might have envisioned that they would have more synapses than normal, right? Just random speculation. So you already answered Connor's question, which was also my question. Like, what would you speculate whether there's more or less in a normal human? Then that your answer made me start thinking about synaptic strength. So did I miss you talking about synaptic strength, or?
No,
okay.
You didn't miss. I mean, the synaptic strength, actually, we see is exactly the same distribution and potency in mouse and in human tissue in my hands. So I think synapses seem to be the same size. I don't see any sort of massive increase in strength in the human tissue. What the difference between epileptic and non-epileptic tissue is, we would have to measure those patients, but of course, it's difficult to do this. Yeah. So, I mean, we have to see.
I wonder if it would be worth, because you need more work, testing an epileptic mouse and a normal mouse, right? Because then, I mean, that would be pure extrapolation, but might give you a direction to go while you're waiting for those, rare samples.
Yeah, we will probably do this actually. The thing is, I mean, these epileptic mouse models I try and avoid, because I try and avoid any mouse models that look like they're having a horrendous time. But I think it's something that we will do a limited data set on to see. Yeah, it's a very good point.
Great, thank you, great talk.
Cheers.
Right, did we have a question from the audience you'd like to have?
Yeah, let me okay, David, I'm should be on screen now. Okay, you could go ahead, David.
Right. Yes, it's really amazing to see how much it's progressed. It's difficult to take in now that it's 45 years since we first pulled a patch-pipette and recorded some single channels. And I can remember in Heidelberg, Bert Sackmann getting 3 connected neurons, or one of his postdocs doing it at least. Now you have 8. That's pretty amazing. But would I be right in saying that at the time of the decade of the brain, we were promised that memory would be understood by the end of the decade? But in fact, and I can remember a few things from when I was, well, the earliest memory I have was when I was 4. I'm now going to be 89 in a few weeks. And I don't think we have any idea, do we, how memory can persist that long? None at all. Be honest.
I would be inclined to agree with you. I would actually also be inclined to say that I don't think we're going to understand the brain by the time I'm done with neuroscience. So I'm sort of see it as there's lots of interesting puzzles to explore, and we'll see what happens. But yeah, we'll see. No, I mean, I guess everything we do is sort of looking at little pieces of possibilities, but it's very difficult to say, look, this is exactly the mechanism. I definitely would agree with you. I'm not going to say that we know it. And that means also that there's plenty of things left to patch. So
yes, indeed. Thank you
as
very
long
much.
as there's things left to patch, I'm a happy man.
Thank you.
Keep you off the streets. Back to the chat if we can. Carlos Obejero Paz asks, how long are these experiments and are there changes with time? I think on a shorter time scale, not years.
With time in terms of, so the slices for like human slices, for example, actually last better than mouse slices in many people's hands. People have used acute slices of humans for up to 48 hours, even 72 hours. We don't have that large tissue blocks, so we'd actually do a length of recording that's very similar to mice. We would record maybe a bit longer, up to 12 hours or so. In terms of the changes over time, I would say that perhaps the first few slices have slightly more activity than the last spontaneous activity than the last slices, but aside from that, I mean, there's really not so much difference over time.
John Gargus makes a point that I think you would agree with. Many of the patients may have at least had seizure gene panels done already, and you should certainly try and correlate this, which...
Yeah, absolutely. I think, this has been sort of like, this was the first time that human slices have been done here in this lab. It's the first time we've had this collaboration. So we're also trying to explore what is even possible to get data-wise and what to implement. And that is, especially if this was going to be a long-term project, would be fantastic to do. Particularly also other things for like memory tests, for example, before and after surgery and these things, and also relating the data we see to whether or not the surgery was successful, whether or not they're then seizure-free would be interesting. So I agree totally. It's just something that we have to integrate with the hospital. Yeah.
I'll come to you next, Layla, I promise. We've got a question from Malka Cohen Armon, who asks, can you do your experiments in PARP1 knockout mice? They have no long-term memory.
We absolutely could. I mean, there's lots of different mouse models. It would be interesting to look at the structure of the circuit in. And so we absolutely could do, we just don't have the mice here. So, yeah.
Layla, would you like to go?
Jake, that was a really, really, really interesting talk. So I had a question about the first part of it, about the two populations of neurons that you find in the mouse and the CA3. So what do you have any idea? And I apologize if this is super naive question, but do you have any idea of what drives, sort of their spatial destiny, the two different populations?
Yeah. That's a very interesting question that we definitely want to explore, which, I mean, it has been shown much more better characterized in CA1, this deep and superficial different cell types. So in CA1, there's deep cells and superficial cells that have been talked about as two populations for a long time. And it's also shown that the development and the time when the neurons are born, definitely in the cortex and also in CA1, that you have this layering whereby the deepest cells are born first. So when you've got progenitors, creating neurons, the first ones are the deep neurons, and then later on the superficial neurons. And so I would imagine this is at least playing some role in the layering and the production of cells, but also I think there will be some genetic component, which is also probably created from development in some way, whereby there will be a genetic difference in the two populations. But this we're not sure of. But
is there any sort of obvious morphological features in the two?
Yes, there's quite a few. I mean, like the branch pattern is quite stereotypically different between the two. So, I mean, you can even tell by eye. The somas are quite a little bit they're a little bit smaller for the deep neurons than the superficial neurons. So there are features that you could classify on purely based on morphology. But we've done it functionally because, of course, we're looking mainly functionally. Yeah. So yeah, there's lots of ways to explore it.
Cool. Thanks.
We're past the hour, but I might just use my privilege as the host for one last question. On Layla's, to build on that, I'm wondering about like what's the stereotypical anatomical nature across the sub-region, right? So with your 8 neuron recordings, can you tell in different parts of the CA3
yes.
network that there's differences? Do you have that resolution yet?
Yeah, absolutely. I mean, so we've done them moving across. I mean, and there's a lot of work done by a lot of other people looking at like the part of the CA3 closest to the dentate gyrus versus closest to CA1, and the properties change. Also, the sort of function in vivo is thought to change. We see a very clear difference with the cells closest to the dentate gyrus are more active. They have different morphology, and I think they behave sort of weird in my hands. So most of the things we've done is focused on the more distal part where things form these two layers. But certainly there's a lot more heterogeneity even within CA3 to explore. I'm sure there's not just 2 types of cells, there's going to be many more. And breaking that down and understanding what it means is going to be plenty of work to do. Yeah.
Fantastic. I think we should finish on that note. Thank you, Jake. That was a wonderful and thought-provoking seminar. Thank you, everyone, for attending. If you have any further questions for Jake, I'm sure you can reach out using the contact on the slide. And we'll see you all next month. So thank you, everyone, for joining.
Thank you very much. Cheers.
Thank you.